Do you think your salad is just greens?
Beneath those crisp leaves, there's a hidden world you never knew existed, and we're going to explore it. AI, scan the salad bowl. We're going to be studying this today.
Scanning. Initiated. Scanning completed.
Let's go to the microscope. AI, tell me what we're going to be looking at today.
Okay, after analysing your salad bowl, I found you have a good variety of Lactuca sativa, most commonly known as lettuce, in your bowl, but the two ones I recommend looking at for observation are the green leaf lettuce and the Lollo rosa, or Lolla rosa, to observe a lettuce with different pigments. Since you want to start with studying plant cell structures, I recommend using the Lolla rosa, since the red pigments will allow you to distinguish the cell structures more easily.
Perfect. You know what? Play something appropriate for the moment.
Playing now, Mum. Your favourite station on Pandora.
Oh, yeah. Where's my salad?
You left it on the end table by your favourite chairman.
Oh. Thank you. Alright, let's see what we find. Starting from the bottom surface, I can see the stomata, tiny pores scattered across the lower epidermis, with guard cells surrounding them. As I adjust the focus deeper, I notice the spongy mesophyll cells, which appear pink due to the anthocyanin pigments present in the leaf. Shifting focus even deeper, I catch a glimpse of the palisade mesophyll cells, which are tightly packed and full of chloroplasts. It's amazing how much goes on in this tiny piece of lettuce, but we're gonna go in deeper, and for that we need a special stain. Remember the stomata I mentioned earlier? Now we're getting a closer look at these fascinating structures. Stomata are tiny pores found on the underside of the leaf, and they play a vital role in the plant's life. Their primary function is gas exchange. The plant takes in carbon dioxide from the air through these pores, which is then used in photosynthesis, the process that converts light into food for the plant. In turn, the plant releases oxygen, which is essential for life on Earth. Surrounding each stoma are kidney-like guard cells, which act like gates, controlling the opening and closing of the stomata. When the plant has plenty of water, the guard cells swell, causing the stoma to open wide, allowing carbon dioxide in and oxygen out. However, when the plant is in a dry environment, or if it's losing too much water, the guard cells shrink, closing the stoma to conserve water. This process helps the plant manage its internal water levels through a process called transpiration. Water vapor is released from the stomata, cooling the plant and helping to maintain its water balance. What's amazing is that the stomata are able to adjust in response to changing conditions, like light levels, temperature, and humidity. On a hot day, for example, the plant will close most of its stomata to minimize water loss, while at night or in cooler conditions, they might open up to allow gas exchange when water isn't being lost as rapidly. It's this delicate balance that helps the plant survive and thrive. Now we're looking at the mesophyll spongy cells. These cells, like most plant cells, have a nucleus. which is the one that controls the cell's functions and contains its DNA. Interestingly, the position of the nucleus within these cells changes with light conditions. Under dark conditions, the nuclei in both epidermal and mesophyll cells shift to a basal-central position, the dark position, centrally located within the cell. Under light conditions, however, the nuclei move along the anticlinal walls, the light position, aligning closer to the cell's outer edges. This movement may help the cells optimize photosynthesis and protect the nucleus under different environmental conditions. Now we're looking at the palisade cells. These are elongated, tightly packed cells just beneath the upper epidermis of the leaf in the mesophyll layer. They're packed with chloroplasts, the organelles responsible for photosynthesis. Positioned to capture the most light, these cells play a key role in converting sunlight into energy, making them essential for the plant's growth and survival. Now, I wanted to look a little closer and see if I could find some of the hidden parts of the cell, and for this I used another type of stain. Based on my research, Lugol's iodine is commonly used to detect starches. However, in some circumstances, it can be very useful for revealing the nuclei of cells. This stain worked perfectly for highlighting the nuclei in the palisade cells. The nucleus is the control center of the cell, housing the genetic material and regulating activities like growth and reproduction. In this footage, I also observe a small dot on each side of the nucleus, but I'm not entirely sure what these are. If you pay real close attention, you will see some dots inside the cytoplasm of this cell, which I am not quite sure if they are peroxisomes or something else. If you know, leave a comment below. Also, if you found something that I didn't mention in this video, feel free to leave your comment as well. Now we're going to talk about something that I initially thought was some sort of microorganism or bacteria moving around. But as I did more research, I learned that there's something called Brownian motion, which is very common in the microscopic world. This is the random movement of tiny particles suspended in a fluid like water or air, and is caused by collisions with the fluid's molecules. This motion was first observed by botanist Robert Brown in 1827, who noticed pollen grains in water jittering around in unpredictable patterns. The particles move in zigzag because they're constantly being bumped by molecules in the surrounding fluid, which move at high speeds due to thermal energy. In 1905, Albert Einstein provided a mathematical explanation of Brownian motion, showing that it's a result of molecular motion, which helped confirm the existence of atoms and molecules. Brownian motion is essential in physics, chemistry, and biology, and it explains the diffusion of substances, such as how oxygen spreads in water or nutrients move in cells. This phenomenon illustrates how microscopic forces and molecular energy contribute to the movement and distribution of particles in fluids. Isn't it amazing everything you can learn from a piece of lettuce? Now let's talk about Nastinovia ribosnegri, most commonly known as lettuce aphids. Lettuce aphids can cause significant damage by deforming leaves and stunting plant growth. They also produce a sticky substance called honeydew, which can attract mold and other pests. Furthermore, lettuce aphids are vectors for plant viruses, potentially spreading diseases like the lettuce mosaic virus. Managing lettuce aphids can be challenging. Farmers often use integrated pest management strategies, including introducing natural predators like ladybugs and lacewings, or selecting aphid-resistant lettuce varieties. Organic farmers may also use insecticidal soaps or horticultural oils, though these are generally less effective. In commercial production, careful inspection and crop rotation can also help manage aphid populations and reduce their impact on lettuce crops. If you eat one of these in your salad, don't worry, lettuce aphids are harmless to humans, but you do want to wash your lettuce well. The last thing I observed on this piece of lettuce is that it was covered on white dots. And as I did more research, I found out that there are two types of mildew that can affect lettuce. One is called powdery mildew and the other one is downy mildew. The one I see here is most likely downy mildew. Downy mildew in lettuce is a fungal-like disease caused by Perennia lactici, which appears as yellow spots on the upper leaves and white fuzzy mold on the underside. It thrives in cool, moist conditions and spreads via spores carried by wind, water, or contaminated tools. When the spores land on lettuce leaves, especially in damp conditions, they infect the plant, leading to reduced growth and potential crop loss. Managing downy mildew involves reducing moisture on leaves, improving air circulation, and using resistant lettuce varieties when possible. Eating lettuce infected with downy mildew isn't generally harmful to humans. However, it can affect the taste, texture, and overall quality of the lettuce, making it less appealing. While downy mildew itself isn't toxic, the compromised plant tissue can be more susceptible to secondary bacterial infections, which could pose a risk if not washed thoroughly. It's usually best to discard visibly affected leaves and consume only healthy looking portions. Now that was it for this video. I hope you enjoyed it, learned a lot, and I'm gonna see you on the next one.